Hello and welcome to M.D.Zafar lectures, where I try my best to make medicine fun to learn. If you are new to my YouTube channel, kindly subscribe and share my channel with your friends as well. So today we are going to talk about the hydrochloric acid, which is one of the most important gastric secretion. Before going into detail of hydrochloric acid and its mechanisms, we will take an overview of different types of cells which are present in different parts of the stomach and how they play their role in the functioning of the stomach. So first of all, when the food is entered into the stomach, it will distend the stomach. Now this distension is a stimulatory factor for the G cells present in the gastric antrum to produce gastrin. This gastrin will travel towards the body and fundus where the parietal cells are present, and it will stimulate them to produce HCl. Parietal cells are the cells which produces intrinsic factor and HCl, but gastrin stimulation only produces HCl. So remember that. Then gastrin also acts on the enterochromaffin-like cells to produce histamine. Histamine again acts on the gastric parietal cell to produce HCl. Now, the chief cells which are present within the body will produce pepsinogen, and HCl that has been produced by the gastric parietal cells will convert this pepsinogen into pepsin. Now, this pepsin will play its final role in starting the protein digestion of the food which has entered into the stomach. So, this is a small flow chart to understand from the entry of the food, how digestion begins, and how different cells play their role in order to potentiate and accomplish the function of the stomach. So now we will talk about the mechanisms by which hydrogen ions and chloride ions are moved from the gastric parietal cell towards the lumen of the stomach for the eventual production of hydrochloric acid. So within each of the gastric parietal cell, carbon dioxide combines with water to produce carbonic acid. This carbonic acid then dissociates into hydrogen and bicarbonate ions. Hydrogen ion is moved from the gastric parietal cell towards the lumen of the stomach by the hydrogen-potassium ATPase pump. Now, this pump also moves one potassium ion from the lumen of the stomach towards the gastric parietal cell. On the other end, the bicarbonate is moved into the blood by the chloride bicarbonate ion channel, and this chloride, which is moved from the blood towards the gastric parietal cell, moves through its own separate channel towards the lumen of the stomach, and eventually the combination of H positive and chloride ion produces hydrochloric acid within the lumen of the stomach. There is also another pump which is very important. This is the sodium potassium pump. It moves sodium from within the parietal cell towards the blood that is outside the cell, and moves potassium from the blood into the gastric parietal cell. The sodium-potassium pump's working is also very important for the final production of HCl because it is of primary importance in maintaining the electrochemical potential so that all the mechanisms can work in harmony for the eventual production of HCl. Now we will talk about the details of the stimulatory and inhibitory factors and their specific mechanisms in the production of hydrogen ion. Now, first of all, we will talk about the stimulatory factors which are mentioned in blue color, and then we will talk about the orange color inhibitory factors. First of all, in the stimulatory factors, we have vagus nerve. Vagus, through its direct pathway by producing acetylcholine, directly acts on the gastric parietal cell through its receptor muscarinic 3 M3 receptor, and by using GQ protein and IP3 and calcium as its second messengers, it stimulates the hydrogen potassium ATPase, and eventually hydrogen moves from within the gastric parietal cell towards the lumen of the stomach. Vagus nerve, on the other end, through an indirect pathway, that is, via gastrin-releasing peptide known as bombacin, stimulates the G cells, which produces gastrin. Gastrin acts through CCK-B receptors, and using GQ protein and IP3 and calcium as second messengers stimulates the hydrogen potassium ATPase pump. Now, gastrin, on the other end, also stimulates enterochromaffin-like cells which will produce histamine. Histamine acts through H2 receptor, that is histamine 2 receptors, and uses a different G protein, that is GS. and also uses a different second messenger, that is cyclic AMP, to stimulate the hydrogen potassium ATPase pump. Now, once we have learned about the stimulatory factors, now we'll talk about some inhibitory factors. The D cells present within the stomach produces somatostatin. Now, this somatostatin on one end inhibits histamine and gastrin, and on the other end also directly acts on the gastric parietal cells, and using the G inhibitory protein and cyclic AMP as second messenger inhibits the hydrogen potassium ATPase pump. Also, we have prostaglandin E2, otherwise known as the gastroprotective prostaglandin. It acts through its specific receptor and using GI or gene inhibitory protein and cyclic AMP as its second messenger, it also inhibits the hydrogen potassium ATPase pump, and eventually less amount of hydrogen will be secreted into the lumen, and overall HCl production will be reduced. So this is the overall picture of the HCl, and thank you very much for listening.